Hello, and welcome to this Evaluate webinar, Gene Editing, Overhyped or Unstoppable Tide. I'm Madeleine Armstrong, News Editor of Evaluate Vantage, the editorial arm of Evaluate Pharma. Before we get started, I'd just like to go over a couple of housekeeping points. We do encourage you to engage with us throughout the webinar. If you have a question you'd like us to answer, please use the Q&A section at the bottom of your screen. We will try and get through as many questions as we can, but if there are any we don't answer, we will follow up with you after the webinar. Our team is also on hand for any technical questions you have. The webinar is being recorded, and we will send you the link to access the on-demand version and a copy of the slides after the event. So with all that out of the way, I would like to introduce our speakers for today. Our first is Sekar Kathiresan, the co-founder and CEO of Verve Therapeutics, a company focused on in vivo editing for cardiovascular disease. Dr. Kathiresan is a cardiologist and scientist who has focused his career on understanding the inherited basis for heart attack. Next, we have Amanda Miklus, Senior Manager, Consulting and Analytics at Citeline. Amanda joined Citeline Consulting in 2018 and works with pharma and biotech clients on disease landscaping and market opportunity assessments. Last but not least is Shardha Millington, consultant at Citeline. Shardha works with pharma and biotech clients, conducting competitive intelligence research and strategic market analysis with a focus on the gene therapy landscape. So, why are we here today? This webinar comes soon after the ASGCT meeting. That's the gene and cell therapy meeting, the big gene and cell therapy meeting, and also soon after the publication of our report, Gene Editing, Overhyped or Unstoppable Tide. This report is free to download, and it features interviews with Verve, Beam, Prime, and Editas, among others, and you can get to it on our website. So the format of this webinar is that I have some questions for the individual speakers, and then we're hoping to have an interactive discussion later. So my first questions are for Shardha. Shardha, you recently attended ASGCT, and I just wondered what the mood was like among the companies present. Just bearing in mind this is a sector that's had maybe more downs and ups on the stock market of late.
Hi. Yeah, great question. So I think the key point to kind of start with, from my perspective anyway, is that it's very clear when you attend the ASGCT conference that the patients come first, regardless of, what's going on in the market. So in the sort of broader scheme of things, it's all about the patients at the end of the day. And, despite any sort of geopolitical concerns that investors might have or sort of market concerns, the cause goes on. Patients continue to be diagnosed with diseases that are fatal or completely life-altering. And so I think that's kind of like the headline point. But as part of that, sort of connected to that, I think there's a huge force to be reckoned with in things like patient advocacy groups, grassroots organizations, nonprofits, and their presence at the conference was very notable. Quite a few of my sessions started off with the patient perspectives. So just thinking of a couple of examples, there was Alison Berendt from the Foundation for Angelman Syndrome Therapeutics, and Kim Nye from the TESS Foundation, talking about SLC13A5, citrate transport disorder. And these were really interesting talks because they basically bring that patient perspective, that lived experience of the diseases, not them themselves living through it, but their children. So it's obviously extremely inspiring. And what was interesting was Kim said that when her child was diagnosed with the condition, the sort of, in her own words, the disease and therapeutic landscape essentially looked like a bunch of zeros and nos, which was really interesting. So that meant kind of zero research conferences, predicted biomarkers, drugs in development, no patient registry, no accessible or free genetic testing and so on, no funding. But now, because of the work that they're able to do, there's 7 research conferences, there is potential for predicted biomarkers, there's a drug in development, patient registry, free and accessible access to genetic testing, and $10 million of of funding for the SLC 13A5 epilepsy. So I think the real feel is there is determination and optimism of what we can achieve at the end of the day, despite everything that's going on, and that has to be the case. And what was also really interesting, I think, was, with gene therapies, the expectation is a cure, or a functional cure. And this is a point that Joy Cavanero from Access Bio brought to the conversation. So she was a keynote speaker, and she sort of provided a really good reminder on the excitement of gene therapies, when she said that there's no other modality where you expect a cure in the first study, and so that's really exciting and promising. So again, I just wanted to emphasize that kind of optimism. But I do want to note also that, there was an interesting talk about VCs and funding, financing, essentially. And they actually made an interesting point, which is how the biotech industry essentially it's always been kind of cyclical. It just kind of so happens that the period from 2011 to 2021-ish was a particularly long up or high period. So, it was the 2020 to 2021 COVID, 2022 particularly difficult, lots of cutbacks in the sort of biotech sector and gene therapy employment. And then now we're kind of trying to pick it back up again. But I think there were some interesting takeaways from what they mentioned on, when you're talking about the markets and kind of investment in gene therapy and gene editing. And the first is, before, a while ago, you kind of just needed a good concept with data and science to back it. But now they sort of suggested that Series B investors are looking at clinical data as a kind of value inflection point, which means that your Series A has to last you just that bit longer. So that's kind of a one point which could be causing some sort of difficulties. There's also sort of leading on from that, a big fear they sort of suggested for Series A or early stage investors is if you're investing in a company and you do sort of 3 years worth of work, you get ready to cross over to Series B, but you don't have enough there, you don't have enough substrate to sort of hand that baton over to a Series B investor. And that's kind of another potential stumbling block. But the final thing I just wanted to mention that was an interesting point was the difference in experience between platform versus asset-focused companies. So asset-focused, they sort of mention you kind of need that in vivo proof of concept, conviction in the indication, and an organized sort of CMC strategy from earlier on. Whereas with platform companies, you can kind of get started with a bit more money because there's more work to do before you get to the stage of focusing on an indication. So those are some interesting perspectives from within the kind of market of financing. But I think overall it was extremely optimistic. Patient advocacy and perspectives were so important. And I think it's also worth noting that, this was the largest ever gathering of the ASGCT conference. So I think that says a lot in of itself. That was my takeaway anyway. I think, people are getting involved. There's more and more research coming out. People are excited to go attend, to network. There were record attendees. I think David Barrett mentioned there were around 7,800 registrants. That includes online, but it's the 10th consecutive year of growth for ASGCT. Memberships are at an all-time high. So I think it was a positive takeaway from my perspective of the attitude.
So it sounds like the climate is quite optimistic, but there are obviously, challenges getting funding, I suppose, and maybe it's a bit different from those days of 2020 when we saw like loads of preclinical companies like getting able to go public. Sounds like it's harder to get hold of cash now.
Yeah, I think it's not necessarily harder, but I think there are sort of more considerations that you need to have. I think your strategies in terms of your like how you're going to scale up manufacturing, sort of CMC strategies, it all needs to be ironed out from an earlier stage. And like I said, like getting ready to have that clinical data early. You know, your early money has to last just that bit longer. So I think it's more just you kind of have to do a lot more maybe nowadays than you did previously, essentially.
Okay. So zooming in on gene editing, because that is the focus of the report and kind of our chat today. So just wondered what you thought were the gene editing highlights from ASGCT, and are there any new technologies or science that you've been particularly excited about from the meeting?
Yeah. So I think there are probably 3 examples that were kind of highlights for me personally from what I attended and from what I saw. So the first off would be exocell data. So this is one of the top abstracts presented. It was presented by Haida Frangel from Children's Hospital TriStar Centennial. Perhaps an obvious pick, maybe. I mean, it's obviously very exciting. Just to recap, so it's a gene editing based technology, so Crispr Cas9 modified CD34 positive human hematopoietic stem and progenitor cells. Joint venture Crispr Therapeutics and Vertex, excitingly awaiting approval. And it's the data presented, I think, was really well received. So it was presented. He presented the CLIMB Thal 111 trial in transfusion-dependent beta thalassemia patients, and also CLIMB SCD 121 in severe sickle cell disease patients. And the kind of key takeaways were all extremely positive. So almost all of the transfusion-dependent beta thalassemia patients treated with the therapy became transfusion-free. So I think that was 42 out of 44 patients. And then 100% of the severe sickle cell disease patients then went on after treatment to be vaso-occlusive crisis-free. Which is obviously extremely exciting. And then across the 75 patients, across the 2 indications, it showed that single dose results in early increases in fetal and total hemoglobin that's durable up to 3 years. And looks like the safety profile is very promising too. Seems consistent with busulfan, myeloablation, and autologous HSC transplantation. So it was exciting to hear him sort of describe it as a potential functional cure, essentially. So yeah, that's number 1. My number 2 would be epigenetic editing. In particular, I'm thinking about Chroma Medicine's presentation. So it seemed like a very well received presentation, not just by myself, and there were loads of questions. People seemed enthused by what they were presenting. Their specific example was epigenetic editors in liver diseases, so it was targeting PCSK9. And basically the aim of their technology, as they sort of phrased it themselves, was to induce a durable change in phenotype, but without having to change the genotype. So their goal here is inducing methylation in the PCSK9 locus at the transcription start site. And the key point here, and this is why I think it's so exciting, no nicks, no cuts, which is sort of a particularly special point, I think, given the wider concerns around gene editing safety and the concerns around double-stranded breaks and potentials for sort of truncated proteins, etc. So it's really interesting because it leverages the endogenous mechanism for regulating gene expression through DNA methylation. So yeah, it's really exciting. So it was a single fusion protein, 3 functional domains. So that's a DNA binding domain and 2 effector domains. With the methylation effector domain, it sort of lays down a methylation pattern that's durable even when the transient structure is gone. And I think the durability is the key point here. So it's very durable in vivo. So I think they found their epigenetic editors had 99% silencing, durable at least 5 months post-dose. And then when you compare that to Cas9, I think Cas9 showed 90% reduction. CRISPR interference showed high reduction, but it was sort of transient. So comparatively, it's looking great. And it's exciting because I think there's potential, they suggest, for a sort of plug and play. You can switch out the PCSK9 targeting guide RNA with one that instead sort of targets the HBV genome. So again, you see that durable reduction of Hep B surface antigen following a single administration. And so, yeah, it's very exciting to see that plug and play approach because, as you know, so I feel like with gene editing, time is of the essence when you're developing a product. So, and then my third one would be new bases, stealth editors. So sort of following on the idea of, safety in gene editing, this is kind of born out of addressing safety concerns that arise from immunogenicity caused by viral delivery systems and bacterial proteins that's sort of still performing in vivo gene editing. So they're really harnessing cellular mechanisms, DNA repair machinery in non-viral delivery, so lipid nanoparticles. So it's just two fully synthetic non-immunogenic components, one being the peptide nucleic acids, and then the second being the oligodonors, which are modified to eliminate immune response and contain the correction. But yeah, so I mean, they tested it in treated human PBMCs and looked at a panel of 5 cytokines to see if they see an immune response, and it looks like there's no problem with cell viability. The results suggest the stealth editors don't elicit an immune response in these cells. So I think they've kind of, like they said themselves, sort of set the stage to moving now to in vivo, which is, again, really exciting. So I think their benefits, in particular, sort of that non-viral delivery, harnessing that cellular mechanism of DNA repair machinery, as they themselves say. And it was interesting hearing them say that it's broadly applicable. I think they noted that it's not only potentially applicable across multiple species, but also potentially applicable to multiple industries like agriculture as well. So yeah, it was a really interesting talk. But those are the 3 highlights for me of Whistle Stop Tour.
Okay, interesting that you've chosen probably the most advanced gene editing project and sort of 2 very early stage projects. But I guess ASGCT is kind of largely very early stage.
Yes,
stuff.
There's lots of preclinical data presented, and so I've having to pick some highlights. I think I've just gone for a bit of a spectrum there. Yeah.
Okay, I mean, I'm seeing a lot of buzz in particular around epigenetic editing, but I guess we'll sort of see how that plays out because there's still quite a lot to prove there, I think. Okay, so maybe just broadly and briefly, safety and delivery have been very big topics. So were there any sessions on those or any comments on those in particular that you want to highlight?
Yeah, I'd say safety in particular was a big one. I think, I mean, I didn't mention this earlier, but Jennifer Doudna actually did a talk. She wasn't there in person, but she was there virtually. David Liu as well, and it was really interesting to hear their perspectives. I know that Dr. Doudna, she said that you need to make this technology more available, but if you're going to do that, and if you're going to do editing in vivo, you need to make sure you're targeting the right thing. And that was basically the general consensus at the conference. Understandably, there's lots of, I think there was lots of focus on ways to improve current technologies and also track off-target edits. So I think on that, I mean, so first of all, one thing to focus on is, advancing detection methods and off-target analysis tools and assays. Just one of which would be a presentation that was done by St. Jude Children's Research Hospital on Change-seq-BE, which basically allows selective sequencing of adenosine base editor modified genomic DNA. So really like understanding the off-target editing profile, essentially. But then the second component is then improving specificity of editing tools. And I think there was lots of interesting data presented. Some examples are, Poseida have their Cas Clover platform, which I believe is based on a fusion protein that's only active when dimerized. And I think they reported that to have high fidelity and specificity, and actually combined with their LNPs has a favorable safety profile and can be redosed. And then also another example, I think, would be Metagenomi. So they presented a new type 5 CRISPR system with potent editing activity in mice and non-human primates. So it showed higher specificity than Cas9, a type 2 system, use smaller guide RNA and smaller proteins. So, there was lots of new things presented, I think, in of itself. So I just mentioned, the plug and play, no nicks, no cuts, epigenetic editing. I think that speaks to, the movement towards trying to think of these safer approaches. Again, the stealth editors, when you're talking about immunogenicity. But I would say it was interesting, the amount of discussion around a kind of balance between focusing between the kind of safety and the need to get this therapy into patients. So I'm just thinking, so there was an interesting talk that had quite an impression on me. It was from Fyodor Urnov from UC Berkeley. And he was talking about, you need to really reduce timelines from the end-to-end gene editing process. And I think he sort of said there are enough what-ifs to never edit anyone ever, which I thought was really interesting. And I think he was sort of saying, if you have to edit people to know how to safely edit people, but obviously there's a balance. But it was just interesting to see that balance come up a couple of times in the conference in relation to gene editing and, the risk of editing versus the risk of doing nothing and then living with the consequences of the genetic diseases in question. I know Kyle Cromer from UCSF also mentioned this in his talk on CRISPR off-target activity and mitigation strategies. I think he kind of said you have to balance the risks with the known risks of doing nothing. And the point is you learn something new every time a patient is dosed. But so those are the kinds of discussions that I saw coming out in the conference. It was interesting to see the mix.
Okay. Well, maybe now is a good time to bring in Sek, because Verve is developing what could be the first in vivo base edited project to get into the clinic. But for those of you who aren't familiar with the story, the project got put on FDA clinical hold last year, and there were sort of questions at the time around off-target editing. So it's probably a good time to bring you in. Just wondered, at your last update at your first quarter, still sounds like you haven't spoken to the FDA yet. So just wondering what the holdup is and if you're waiting for these Heart-1 data before you speak to the FDA.
Thank you very much, Madeleine, for the opportunity to be on this webinar. Let me take a step back and just kind of catch people up on what we're trying to accomplish here at Verve before jumping into your question regarding different regulatory authorities. So at Verve, we're developing one-and-done gene editing medicines for cardiovascular disease. Our lead investigational medicine is called Verve 101. It's designed to be a one-time intravenous infusion that leads to permanent lowering of blood LDL cholesterol as a treatment for a disease called familial hypercholesterolemia, or abbreviated FH. This disease, FH, is a morbid genetic disease characterized by high cholesterol and heart attack at a young age. It affects about 3 million people in the US and Europe, and about 30 million people worldwide. Now, our medicine is designed to make a single spelling change. Shardha described base editing. So this is a base editor, and the change is supposed to happen in the DNA of liver cells to turn off a cholesterol-raising gene called PCSK9. And so in some sense, I tend to describe this as really surgery. It's a molecular surgery, a permanent change intended to lead to lifelong benefit. And so we've taken this approach to a number of different regulators, New Zealand, UK, and US last year. in order to be able to dose a first patient with our medicine. And as you know, regulators typically assess a new medicine through the lens of risk and benefit. And with our global regulatory strategy, we've gotten our first 2 clearances to test the medicine from New Zealand and UK. And we've been dosing patients with FH in those 2 countries and expect to present clinical data on dose response across 4 dose levels in the second half of this year. And as you said, this will be, I think, the first demonstration of in vivo base editing in humans. And in terms of the third country we submitted to, the US, we're working with the FDA right now to obtain clearance in the US to dose the first patient in the US. Our discussions with the FDA have been very collaborative. We're continuing to engage with them in both with teleconference and written communication. And I think what I can say is that all the regulators, I think, recognize the unmet need that we're trying to address, which can be summarized as right now less than 5% of patients with FH actually are at LDL goal. That's despite the fact that there are 3 pills right now and 3 injections available to lower cholesterol. So the regulatory authorities recognize this unmet need and that our approach of a one and done has the potential to be a definitive genetic treatment for this genetic disease. And I think, as I've described before, with respect to the FDA, our impression is it's a matter of when, not if, in terms of getting to that clearance.
Okay, and will that data from New Zealand and the UK be included in your response to the FDA?
We do expect to include the existing human data, and that's one of the things the FDA asked for. They basically said, we recognize the unmet need. We don't have any issues with the clinical trial population or the clinical trial protocol. We're fine with the CMC. There were no hold issues on the CMC. The hold issues were really all preclinical questions, and we're working with them to address those. In addition, they said, look, we know you have a trial going ex-US. So when you're ready to submit your response, please provide whatever clinical data you have available from New Zealand and UK at the time. And that's what we plan to do.
Okay. And we've spoken about this before, and you alluded to it just there, but there's been questions kind of about the interplay between, gene editing being used in rare versus common diseases, maybe with regulators favoring diseases where there are no other options, but you don't believe that is the case here. Is that correct? You said there's no...
And I think that it's our experience with those 3 regulators, and 2 of them, they've cleared it, and we are dosing patients right now for heterozygous FH. And then in the US, of the 3 review areas for an IND, we did not get any hold issues from the clinical trial protocol. So that I think what supports my thought that it's really not about common versus rare. And of course, we're focusing on a genetic subset of the common disease, or that there should be no treatment options available for this approach to be first taken to patients. The second supporting evidence on that point is, of course, Intellia. Intellia's FDA clearance for a phase 2 was in hereditary angioedema, where there are actually lots of treatment options currently available. In fact, very effective ones, and monoclonal antibodies. And with all that, of course, they've been able to get a clearance for a phase 2 IND.
Okay, I'm glad you mentioned Intellia, because one of my questions is around their technology. So I think when we spoke earlier this year for my reports, you said you thought the caution from the FDA might be around lipid nanoparticle delivery, but the Intellia project is also delivered via LNP. So do you think it's more caution around base editing versus CRISPR?
There's an open IND for base editing. So I don't think it's actually related per se to base editing versus CRISPR Cas9. I think that, look, the different agencies have different approaches to a new technology. I think the FDA's approach has been very deliberate with this new technology. What we were asking for was the ability to dose a first patient, really a phase 1 IND. They reviewed our protocol, have some additional questions, and we're working with them to really address those questions. So I think it's been a very collaborative process. Like I said, I'm confident that we'll be able to work our way through this. I think in the meantime, as you've noted before, Intellia did go and get a clearance for a phase 2 after performing a phase 1 clinical study in New Zealand and UK for about 2 years. And then they took that phase 1 patient data to the US this year and got an IND clearance for a phase 2. So that's certainly one approach that we could have taken. But again, we chose to engage with the FDA at a phase 1 at the earliest stages of development, because we really want to understand, not only for this product, but for all our future products, what the bar is going to be for a phase 1 clearance in the US.
Okay. So another question arising from your first quarter results is that you unveiled Verve 102, which also edits PCSK9, but it uses a GalNAc LNP delivery technology. So just wondering what that means for Verve 101, and there's been sort of an idea that Verve 102 could be regarded as like a hedge. Is that accurate?
I think we're very committed to the PCSK9 target. And with the introduction of 102, which utilizes our own proprietary GalNAc lipid nanoparticle, we actually have just more optionality for patients. Ultimately, we want to choose one of the two to take forward into later stages of development, so phase 3. And we're going to let the human clinical data that emerges from Verve 101 and 102 to guide which we ultimately take forward, which product really is going to be the best for patients to take forward into later stages of development. There are distinct differences between the two products, and the 102 has some potential advantages because of the addition of the targeting ligand, the GalNAc, and those potential advantages are that there could be a bit more potent because this lipid nanoparticle can get into hepatocytes through any of two different receptors, either the LDL receptor or the ASGPR receptor, in contrast to a standard lipid nanoparticle, which only gets in through one receptor. So besides the potential potency advantages, there's also a potential kind of tissue or target organ specificity in terms of the liver, because the two most abundant proteins on the surface of hepatocytes in the liver are LDLR and ASGPR. In fact, the ASGPR receptor, which is the receptor for the GalNAc targeting ligand, is only expressed on hepatocytes in all the cell types in the body. So because of those two potential advantages, we are really taking this product forward into clinical trial initiation in the beginning of next year, Verve 102. And again, as I said, we'll see how each of the two products perform in patients and then pick one to take forward into phase 3 and beyond.
Okay, and you mentioned that the GalNAc LNP is wholly owned. Will that be a consideration when you're deciding what to take forward as well?
Yeah, that's the other potential advantage here. It's that we have a potential business advantage in the fact that this is our own internally developed GalNAc, where we've already been awarded IP, chemical composition of matter IP from the U.S. Patent Office, and so that's definitely a consideration.
Okay, and you previously said that ultimately you want to get to lipid lowering levels seen with inclisiran. Leqvio. So I guess watch this space to see which one can do that, or maybe more, it sounds like.
Yeah, I think that our goal is to get the LDL as low as possible for as long as possible. And certainly we've been able to pretty much completely turn off the gene in monkey, in non-human primates. And when you do that in the liver of non-human primates, you're getting about a 90% reduction in blood PCSK9 and about a 70% reduction in LDL. Now that's, I think, we'll see how we can how the translation goes from monkey to human in terms of the dose response. And and certainly would love to see a dramatic response like that. But then there's the concept of what's a minimum viable commercial product. And we think a minimum viable commercial product would be one that matches inclisiran in terms of its efficacy. And of course, the differentiation for Verve 101 would be the durability, the fact that ours would be optimally a one and done, so a lifetime of benefit, whereas inclisiran is usually several months. So in terms of, then that brings us to what are the data for efficacy of inclisiran in HeFH patients, in heterozygous FH patients. So in a phase 3 called Orion 9, so this is the registration trial for inclisiran in HeFH patients, inclisiran lowered blood PCSK9 compared to baseline by about 60%, and it lowered blood LDL by about 40% compared to baseline. And so this is, I think, a reasonable starting point as to what might be a minimum viable commercial product.
Okay. And how do you feel about the threat of new technologies coming behind? So Shardha talked about epigenetic editing of PCSK9. So that seems something that could threaten you eventually, although does sound like it's quite far away from reality at the moment.
Yeah, I mean, this is, FH affects 30 million people in the world. Atherosclerotic cardiovascular disease, so garden variety heart attack, which would be the next indication we try to take this approach to, affects probably 200 million people worldwide. So there are lots of patients that there's an unmet need, and I think there's room for multiple technologies to take a crack at this. And I think there's reason to be excited about new technologies, but as you know, there's also reason to be cautious. I think for epigenetic editing, I think the efficacy is a starting point and durability is a starting point, but then they're going to face the same questions that others have faced as to off-target methylation. So, for example, might there be other genes that are methylated and turned off that you may not want to turn off? And how are you going to detect that? In what cell types are you going to detect that? What are the approaches? Has there been standardization of the evaluation of off-target methylation, like there has been standardization now of evaluation of off-target DNA editing? So there's a lot of work to do but a lot of potential for each of these new technologies.
Okay. Well, so maybe time to bring in Amanda now and talk about some of the deals we've seen, because I know Verve has also been involved in some of these deals, which you're going to come onto later as well. So Amanda, there's still been quite a lot of deals recently in the gene editing space, even despite the kind of suffering of some of these companies on the market. So what kind of appetite do you see among some of the bigger players for the technologies we've talked about?
Firstly, thanks, Madeleine, for having me on this webinar. And so when you consider the partnership deal-making landscape for gene editing companies over the past decade or so, the answer is there is still a very big appetite for gene editing, despite these difficult public markets. And I think it's the draw of this technology and the potential that it has, speaking to what Sek was mentioning, addressing these high unmet needs, curing diseases, and providing lifelong benefits. So there is a big interest from large cap pharma companies, top tier biotech companies consistently over the past several years. And we see broad representation from many different big pharma companies, top biotech companies, including Regeneron, Gilead's Kite division, Bristol Myers Squibb, Vertex, Sanofi, and just recently Novo Nordisk signed a deal with Life Edit. And one of the key trends that I've been following is the largest deals, in terms of the largest dollar values that we've seen attributed to these deals signed in the recent couple of years, is around the subset of in vivo gene editing, which Shardha talked a lot about, the findings from the recent ASGCT conference. And some of the biggest players are involved in these in vivo gene editing deals. So, for example, Pfizer has a $1.7 billion deal with Beam. It's around liver disease, muscle and CNS diseases. Bayer has an agreement with Mammoth worth $1 billion around liver-targeted diseases. Precision Biosciences, which Shardha mentioned, there's favorable safety with their technology. They have a deal with Novartis. Poseida has a nearly $4 billion agreement with Takeda that includes Poseida's Cas-Clover gene editing system. And just recently, Scribe signed an agreement with Eli Lilly's Prevail around CRISPR X editing, their CRISPR X editing platform. And as I mentioned, Vertex is very active in this area and has an agreement with Verve worth almost $500 million for a liver disease program. So there's a lot of attention here in the in vivo gene editing space, and big pharma companies, large cap pharma companies are definitely investing. And we're lucky enough to have one of these deal makers on the panel. So it would be great to get Sek's input on and give a little color behind their deal with Vertex.
Glad to do so. Thank you so much. Yeah, so we're focused on in vivo liver gene editing and cardiovascular disease. So that's kind of what the company is built around. But Vertex approached us now almost a year and a half ago, I guess, and because they had seen our GalNAc lipid nanoparticle technology and the efficiency with which we're getting in the liver in non-human primates. And specifically, data that we'd released for our second medicine target called ANGPTL3. ANGPTL3 is a gene that normally raises cholesterol and triglycerides. We'd shown that you can turn off this gene. We could turn off this gene in the liver of non-human primates and bring the blood ANGPTL3 level down by about 96%. Now, the blood level is almost entirely made. In fact, it's entirely made just by hepatocytes. So the fact that we could bring it down by 96% meant that we were editing basically every hepatocyte in the liver. That's like 200 million cells in an adult liver. So really very high efficiency editing with GalNAc lipid nanoparticle. They saw that and they said, oh, maybe we should really think through this delivery approach for the project that they were involved in, the target they were interested in. So that led to a series of conversations, and then ultimately we announced a deal last year that I think the total initial consideration was about 60 million, combination of cash and equity, and then there's some downstream milestones and royalties, as well as an ability for us to opt in potentially to a profit share for a 60-40 split down the road. And importantly, the deal is structured in a way that they're paying for all the research costs, all of the next couple of years of research costs, few years of research costs till to phase one. And so that, of course, helps a company like ours where we have this focus on cardiovascular disease, lots of other projects, and then now we have a partner to kind of to help us take this on. So it's been very productive so far. And the thing that I'll note is that this is not cardiovascular disease target. So it's actually a genetic liver disease target. And we're going to utilize our GalNAc LNP delivery technology. But then the question becomes, what's the editor that we'll use? And here, I think we've taken the approach that we've taken to all of our targets, which is we're technology flexible. We're not focused on one technology per se for all of our targets. Rather, we take the target and say, all right, for the edit that we want to make, what's the best approach? Is it standard CRISPR-Cas9? Is it base editing? Is it some other form of editing? And what we've decided for this project is it is not going to be standard Cas9. It's not actually base editing, but rather it's a custom editor that we're developing bespoke for this target. So that I think kind of speaks to the evolution of our company from one intensely focused on a couple of targets and a couple of technologies to one where we're now, I think, going to have a pretty full technology stack in terms of mRNA capability, guide capability, delivery system, as well as an editor development component.
Yeah, thanks, I think this is just a perfect example of what we're seeing is deal making being driven by good data, strong science, and again, back to your question, Madeleine, when that evidence is there, we see big pharma companies ready to make these deals with gene editing players.
Okay. Interesting. So, Amanda, what sort of trends are you seeing then? I know you've done an analysis of deal values.
Yeah, thanks, Madeleine. And if, we do have a couple of slides. If we could pull up the first slide. So I think, as I said, for the right asset, big pharma will pay. They're looking for strong data, but they're also looking for a strong fit with their strategic portfolio at the end of the day. So in looking at average total deal values and average upfronts, we're seeing a couple of different stories here. So the chart on the screen shows the average total deal value for gene editing deals going back to 2014. And you'll notice there's a couple of different peaks here. There's a peak in the 2015 to 2016 time period, and then again in 2020. And during these two peaks, there were quite a few deals that had very large deal values, one of which in 2015 was Crispr and Vertex, which Shardha talked about earlier, the partnership that eventually has produced Exocell. So that deal was worth $5 billion. It's been expanded, so the total deal value is now at $5 billion. And then in 2016, we also saw an agreement between Regeneron and Intellia. That was an in vivo gene editing deal, actually. And also Biogen and UPenn have a $2 billion agreement that includes a gene editing component. And then in 2020, that peak was really, that average is really boosted by a deal between Vertex and Affinia, which is worth almost $5 billion. And then a deal between Repair and Bristol Myers Squibb around synthetic lethality involving CRISPR for a total value of $3 billion. And another thing that I thought was interesting is that some of the key events that have happened in the gene editing sector, we can kind of see correlations between those events and some of these peaks and troughs. So, for example, between 2011 and 2014, we saw the founding of some of the key gene editing players, CRISPR Therapeutics, Intellia, Caribou, Editas, for example. So I think what we're seeing is the foundation of these companies, them getting a lot of venture financing, and then actually getting big company partnerships. In 2014, the USPTO granted Feng Zhang and the Broad Institute their foundational CRISPR-Cas9 patents. In 2019, we saw the publication of what was then the new DNA technique, editing technique called prime editing. And then in 2020, the first patient received gene editing therapy with CRISPR directly into the body at Oregon Health and Science University. And also in 2020, the Nobel Prize in Chemistry was awarded to Charpentier and Doudna. So we see some of these key events happening. And then also it drums up a lot of deal-making interest. One thing to note, though, which is very evident, is that since 2020, we see this downward trend in average deal values declining from 1.6 billion in 2020. And in 2023 to date, it's at around 845 million. So I think the question for me and the question for a lot of us is, are we seeing a correction or a return to kind of normal levels, or is this going to continue to be a cyclical issue? And if we go to the next slide, which is average total upfront values, there's a slightly different story, as I mentioned here. So if we kind of remove or disregard the outlier of 2015, which again includes that very large deal by CRISPR and Vertex with where you have an average 333 million upfront, which includes the 900 million upfront total that is part of that Vertex and CRISPR deal. If you exclude this outlier, what you can see actually is that generally upfront payments are trending upward year over year since 2019. So, for example, in 2014, the average was around $30 million for a gene editing deal. Then shift forward to between 2017 and 2021, most years hovered around $50 to $60 million upfront. But in 2022, we saw the average upfront rise to almost $100 million, so $95 million. And even to date in 2023, as of May, the average is $63 million. And so overall, I think when you look at the upfront and then when you consider the total deal value, I think what we're seeing is that companies are seeing valuations increase. They're seeing more money upfront on average over the past few years. So more front end loaded deals, and the decline in total average deal value that we saw in the first chart, that could potentially indicate that maybe the milestones are decreasing. So potentially we might be seeing a higher confidence in this technology. So I think that's the story that we are seeing, is that the valuations are increasing, at least on the upfront side in gene editing deals.
Okay, I'll be interested to see how that evolves over the next couple of years, especially that first chart, as you said, to see if it's a correction or if it's kind of a return to what we previously saw. Okay, well, we've been talking for nearly an hour now. So unfortunately, I don't think we're going to have any time for questions from the audience. There's just been too much to talk about. We need another webinar, I think. So thank you, all three of you, for a really enlightening discussion. Just to wrap up, thank you all for everyone who joined us, and thanks for submitting any questions. We will be in touch to answer any or all of them that we didn't manage to cover in our discussion. A big thank you to our panel of speakers today for your insights and thoughts on this topic. And if anyone would like to speak to us about this further, please drop a note into the Q&A and we will reach out to you shortly. And we'll send the full recording of the webinar out soon, along with a link to download the slides and the report. Looking forward to seeing you at the next Evaluate webinar.
Thank you.
Thank you.
Thanks. Thank you.